Anyone? Don't see anyone. Just joking. And if y'all can think of some questions for Jennifer, that's fun for her too. Questions from anybody? Otherwise, I'm going to ask you, Sarah. So they did this in a person?
Yeah. Well, they did it on.
Oh, okay, like a rat or something. So they said it's a gel. So did they like rub it on the head or something? Yeah. So I think it's. Do you remember how we talked about the blood-brain barrier? That's just a fancy word for it. Huh. That's crazy. Yeah, because it's hard to get neurons to grow. In general. All right. Cool. Thanks. I put your extra credit in there. Just do it. Sweet. I could use some of that gel. Give me some more brain cells. All right, so for today, we're going to finish chapter 15 and then basically just go through the study guide and have you ask me questions about it. Which ones you want to go over? Turn this on. I don't know. You're going to have to tell me where we are. I know we have quite a wait. We have a decent amount of stuff to go through. Well, maybe not. I don't remember. Okay. All right. Which is not too much. Eight. Oh, I remember. We still need to do that one. Good find copy. All right. So, actually, the main thing we had to work on is group question number 8 on your worksheet from part 2. So. This will be actually a really good exam review for you. And it's to compare sympathetic versus parasympathetic. And I've got you a nice little table to fill in. And the reason I put it that way is because if you write something in this box over here, then you need to write something that's different in the other box. but it's kind of the same. So for example, don't write, sympathetic tells the heart to beat faster, parasympathetic tells the pancreas to do this. So those don't match, right? So put something about heart here, something about heart here. Same thing, we learned about the different neurotransmitters. Could be comparing those. The locations of the neurons. Basically anything that we went over already. And I've got a few extras, if anybody needs one. Maybe I only have one extra. What? There's some. Hold on, I'll find somewhere. I was just looking at them a minute ago. I thought. Oh, that's the other part. All right, who else wants one? Here's one for you. And Lily, do you want to work with this group over here?
Yeah.
Since no one wanted to. What, this whole table?
I don't know where mine's at. Thank you.
All right. So look back at your notes, look back at whenever. Talk to your group mates. Does anyone want one of these?
I don't know if it's I can't think of the word.
What?
So for this one,
Uh-huh.
like, because I know it said it like the blood flows there.
Yeah.
But how
More
do you-
blood flow.
becomes what? The,
Engorged.
that's engorged. That's what I was trying to think.
Were you at this table just giggling your face off? Yes. At lab.
No.
That group, they were just like laughing so hard about that yesterday during
It's
lab.
not even funny anymore. I'm a CNA. I see way too much of all that crap. I see way too much old man stuff. But no, way too much old man jokes. Yeah, it's.
Yeah, that's sad getting old. That's terrible. So,
Looking for all my stuff and
yeah.
I couldn't find it because it was everywhere.
So if you're not sure where to start, think about how maybe some of the organs respond to those systems.
Are we gonna go over the other study guides?
Yeah,
We're just finishing this up, right?
yeah, because that's a big part of the task, or at least a decent amount. So at least 6 differences. So it could be differences in structure, differences in function. Gotta look back at your notes. You did already. Maybe some of the neurotransmitters, like from 2 through 7, that you answered last time, if you have that.
Okay.
So that this type is columnar.
So if you're gonna put something here, then you should put something there that's opposite.
Okay.
Sorry, I'm really confused though, because now it says it's opposite.
Tuminescence. So that's the
apparently
word.
what it's
I've
called.
never
But
seen
it
that
said
word
for
before.
Sympathetic is the D one, so which means it's for like blood flows away.
Blood
But wouldn't
leaves
you
it.
think it goes towards it during the action?
Yeah.
So sympathetic is when it goes towards it.
I think it's just like
Is it the opposite?
parasympathetic doesn't do anything.
Okay.
Do you know what I mean? It's just this one.
Okay.
Just the sympathetic. And then when there's no sympathetic, then it's just not engorged.
I don't know why I have to be so confused about this. I just want to make sure it's right.
It confuses me that
It says it in the book, so I can.
Yeah, but it's not always true. Like if you look at this table, see it says effective parasympathetic. A lot of the organs say no effect, no effect. Like they're not
Right here it says,
connected.
like one does and one makes it go away.
It says parasympathetic somewhere.
Well, yeah, because it says inhibits dectum, whatever that word is, and then if I click on that word, it says that's when it gets swollen. So wouldn't that be afterwards?
Right, go back.
that's what confuses me too. Is that the
It's like two negatives.
Yeah, like so. This is sympathetic.
Yeah.
Okay. and
Oh, so it-
inhibits that. So it inhibits it to not.
Right.
So then it wouldn't.
So it inhibits D2 in essence, which means
It
engorged.
But this says that's when it leaves it.
Right. So if you stop the
turning-
blood leaving, then you keep it there.
Yeah.
No,
so then
stop
I'm
the blood
wrong.
leaving.
Oh, okay, no, then it's
Stop the
So
blood
it
leaving.
stays
The blood
there.
there.
Keep the blood there.
Okay. I'm
that's
learning too much.
confusing to me though too because it's because it's sympathetic.
I
like,
seriously
does,
just don't
like, the-
understand.
like the fight or flight.
Guys.
You know, like you think like you're more intense.
Like, I was trying to like do some of this, and Ellie's trying to explain to me, like, oh, you know.
No.
He's like, I'll explain the female one to you too. I'm like, no, it's okay. I don't think I got that one.
Well, you've got at least 6.
Yeah, I got all that.
Maybe you can get one for receptors.
Talking about that one. That's what it was.
What about something from these answers about receptors or neurotransmitters?
Yeah, that's all like jumbled in there.
Well, it's all, you got all the answers there. So all you have to do is compare two of them. Right? For which neuron, the pre or post? So make sure when you're putting those down that you do like know what they mean.
Yeah.
So like simplify it or
You would need the pictures, right?
yeah, you could look at the picture thing. So you're talking about the heart rate?
Yes.
You should write
I'll
that.
write that down.
Yeah, because what's heart increase?
I don't know if I'm doing right, but at least increase the alertness and conserve energy.
Yeah.
Neurotransmitter, I think is the same in both. No.
So if you look back at this one, it'll tell you. So it depends on which neuron you're talking about. Because you're right, which neurotransmitter released by the pre, ACH, and then the pre-sympathetic is also ACH.
But it's this,
So I'd write, so it'd be like neurotransmitter ACH by pre-ganglionic for both of them. I mean, technically we were doing differences, but I mean, it's fine to put a similarity on there as long as you remember that it is. Yes, use that table, and then you've got this one. It's got all the responses. Adjust shape of lens for distant vision.
Yeah, increase the breathing percent.
Yeah, because you need more oxygen to survive. Oh, that one's done. You could also think about the structure of those systems. So which of them is like all down the spinal cord? So sympathetic was more spread out along spinal cord, and where was the parasympathetic starting? Yeah, which one had a trunk? Remember? So sympathetic is a trunk. Does parasympathetic? And for you're talking about the sphincters, right?
The what?
For the intestines, the sphincters.
That's what they're called?
Mm-hmm.
How do you spell that? SPHITRS. Mm-hmm. Sphincters. So we have to put that on the test. We can't just put it in testing.
Well, because that's what's relaxing, right?
Oh.
The sphincter in the intestine. It's not like the whole intestine's just relaxed, right? It's like the little gates between the different parts. It's a lot of little words in there. But they're connected together. Well, that's why it's hard,
You know what
CB.
trips me out more than anything? This actually just freaks me out. That my brain does not understand about my brain works.
Well, my brain doesn't understand it either.
Think about that. No, like think about that. That our brains, like they're so freaking complex.
Yeah.
It's amazing that they're so complex that it can't understand it.
But your brain's complex, so it should be able to.
That's what I'm saying. That's my point. That's my point. Like think about that.
Yeah.
You are learning about the thing that makes you learn stuff, and you can't learn that because you don't understand it. That will trick you out. So that set me into like no, just like it was like it was intriguing.
I knew what you'd have down here. Yes.
And just to clarify,
Mm-hmm.
would it be exciting arousal and then explosion because
Mm-hmm. That's correct. And that's one of the few cases where it's like they're both kind of involved at the same time. Usually it's like one or the other.
Right. Okay. What else for the sympathetic? For the eyeball, would it be constricting?
It's the other way around.
Oh, okay.
Because the eyeball, it dilates for sympathetic to let more light in and help you see better.
This one constricts.
Hmm.
Okay. Got it.
Alright. All right, so as long as you've got at least 6 on there, you should be good. And do you think this is going to be on the test? Yes. Do you think it's going to be a short answer question on the test? Yes. So do you think maybe you should make sure you know it? Yes. All right. Which means, it doesn't matter which differences you have as long as you've got some differences. They don't have to be the same as everybody else. I mean, it says 6 on that worksheet. 50? Oh, Jordan wants us to do 15. Okay, so we'll do 15. I haven't decided yet. We'll see. It won't be more than 6 differences. So the last thing we had was an autonomic reflex arc to kind of show you how this all works together. Because you do have parts of the autonomic nervous system that sense things, even though it's not conscious. They are checking up on everything. So your heart rate, how hard it contracts, your blood pressure, all those things are under involuntary control and monitored and kept at a sort of constant rate. So an arc has a sensory neuron, an integrating center, and then motor neurons and an effector. And you'll see how all those work in a second. So we'll use the example of a heart rate reflex. So you have a sensory neuron that senses low blood pressure. That's kind of the first step. It tells, it's a neuron, so it sends its information to the medulla oblongata in the brain to tell it, hey, our heart rate's low. Maybe you should do something about it. And medulla activates the sympathetic nervous system. Because you remember, what does that do to your heart rate? Increase or decrease? It's going to increase it. Now it's not, direct. It has to go through the preganglionic neuron, go through the postganglionic neuron, and then the end of that postganglionic neuron is at the heart, where it sends the little, what does it send out to the heart to tell it to contract faster? What's released from a neuron? Yeah, so a neurotransmitter. So if your heart's beating faster and harder, that leads to higher blood pressure. So you fixed your problem. Yeah. So for exercise, you have some other reflexes that sense low oxygen in the blood. Yeah. Right. Yeah, no, that is cool. Like when you're exercising, it takes a little while for all this to get faster heart rate, faster breathing rate. So yeah, right at first is when you're like really breathing the most heavily. All right, so here's a diagram of that. Same thing. We've got the sensory receptor here in the aorta, senses low blood pressure. It's in purple there. It goes over to the medulla. The medulla decides to do something about it and sends the signal back out through the pre- and post-ganglionic neurons. And it does work both directions. So you can imagine everything opposite too. So high blood pressure turning on parasympathetic nervous system would be the same thing. So it could be high blood pressure coming through the neuron, telling the medulla that there's high blood pressure, activating parasympathetic, pre- and post-ganglionic, sending neurotransmitter to the heart to tell it to beat slower and lower the blood pressure. But kind of the same logic behind it. All right, and just as a reminder, the hypothalamus is what's in charge of deciding whether it's sympathetic or parasympathetic that's activated. So the hypothalamus is the one that decides, the part of the brain that decides whether sympathetic is being activated or parasympathetic. Yeah. So it takes in input from, all different sensory receptors, takes an input from like the conscious part of your brain, the cerebrum, sort of integrates it and decides what to do about it. Because as you saw yesterday, were you able, at least for some of your experiments, were you able to change people's heart rate? Just by them going into that scary dark room over there, or listening to relaxing music for a few. I guess that didn't work perfectly though, right? It's like a couple people it went up. So anyways. All right. So anything about 15 before we do some review? Let's do it in this order. Let's do 15 since we just did it, and then let's go back to 12. So that's probably the most challenging one. Yeah, and I want to tell you for 15 part 2, you could take 2 things off that we didn't go over. Remove. So you can remove P and Q from, yeah. I always think chapter 15 is going to go fast, and then it never does. Yeah, so what do y'all want to hear about from 15?
I'm just still kind of confused with the trunk on the sympathetic nervous system, and it says to compare the structure of them. I know it's probably just like sympathetic.
Yeah, I think the easiest thing is to pull that diagram up and talk about it. Is that on part 1 or just part? No, it's only on part 1. It has the picture. Right. Let's see if we can get both of them. See if we can get both of them side by side. That's really tiny. That's not going to work. We'll just look at one after the other. All right, so in terms of the structure of the 2 systems, what I would probably be remembering is sympathetic basically goes down most of the spinal cord, where the preganglionic neurons start. So you can see them all starting there. And then if you looked at the parasympathetic, you could see that it's starting, those preganglionic neurons start in the brainstem and then way down at the bottom of the spinal cord. And remember, you will have those two diagrams with you for the test. So if you're stuck and you kind of know what that diagram is talking about, you can get lots of answers from there. And then second thing for structure is the sympathetic has that bundle of axons, the sympathetic trunk next to the spinal cord, you know, all the way basically from the skull down to the bottom of the spinal cord. And parasympathetic, we don't have anything like that at all. It's not on there because it doesn't exist. Mm-hmm. Right? Because you don't see any coming out from the middle here.
So they have no roots like they fall in your spinal cord.
Right. Mm-hmm.
Because even after that, that's not even your spinal cord anymore down there.
Right. Not technically, but there are neurons down there. But you're right, it's not the spinal cord itself. Because it stops here. But it's funny because even the book will say that this is coming out of the spinal cord when it's not quite the spinal cord, but that's okay.
And it says that your spinal cord touches.
I know. It's kind of funny. There are still neurons in this part, but it's not technically the spinal cord. It's much thinner. And then which of those has the preganglionic neurons going farther? So it's the para, right? Because for a lot of those, it goes almost all the way to its final destination. The dark, the preganglionic. And then in sympathetic, you know, it goes to that ganglion first. You can see all those ganglions. Or maybe it only goes to the trunk, the preneuron.
So the axons are longer from the parasympathetic. So in the sympathetic, with the axons that go from the spinal cord to that trunk, is like, and then like
A lot of them do.
communicate with a new neuron, and then a new axon comes off of the trunk.
For the ones sort of on the top and the bottom, that's true. But you could also have, remember there was that 4 ways that the sympathetic neurons could go through the trunk. Or I forgot what did I write exactly? How did I write that? The 4 ways that, the 4 ways that it connects with the postganglionic neurons. All right, what else from 15? Part 1 or part 2? So for M, the differences between sympathetic and parasympathetic in terms of type of neurotransmitters and receptors involved, that's questions 2 through 7 on that worksheet we did last time.
I just didn't know how to write.
Okay, let me show you that picture. Yeah, it's on the front page of part 2. Oh, sweet. And you also have this. Does that look familiar? Is that in your book? Yeah. So what you'll need to be able to talk about, first of all, just ignore the sweat gland thing. So the first one at the top is the sympathetic, and the preganglionic neuron sends out ACH, that's the neurotransmitter, and then the postganglionic has a nicotinic receptor. And then the postganglionic sends out norepinephrine, and the effector cell has a adrenergic receptor. That's sympathetic. Parasympathetic down here, the first step's the same, right? The preganglionic releases acetylcholine, and then nicotinic receptor on the post. But then at the target cell, it's acetylcholine released neurotransmitter, and then the muscarinic receptor. So I know that's a lot of big words in there. So maybe you can come up with an easier way to remember it with like a little sentence, or I don't know. Because really, all you'd have to remember, since which part of that's the same? Where the pre meets the post, or where the post meets the effector cell? Right? So if you know that pre to post is ACH and nicotinic, you know that's for both of them. And so then all you have to remember is the difference at the end. Maybe one way, what do you think epinephrine does? So you may know. Okay, that doesn't help, never mind. Well, it's like adrenaline, basically. So it increases your heart rate, breathing rate. But if you don't know if you don't already know that doesn't really help. I don't know, what's an easy way to remember sympathetic is NE? Just that the sympathetic releases norepinephrine. Sympathy for Nebraskans. I heard somebody whispering that. Did you say that, Ann? Did you say that? That's funny because you said that. Okay, so sympathy for Nebraska. There you go. That'll tell you sympathetic. So sympathy for Nebraska. Cool. That'll help a little bit, but otherwise that's just something you have to memorize. No cards, something like that. Which isn't that hard. Yeah, parasympathetic is ACH, but then nicotinic at the postneuron and muscarinic at the effector cell. So you could just memorize that diagram, draw it a bunch of times if you're more visual. Or you could do it in words. No, but sometimes that can help you remember it better. Yeah, we drew it, I think, and then we added the neurotransmitters on it in part 1. Personally, I'm more visual. I would have a lot easier time drawing that picture a few times and remembering it than just remembering words. There's something about like drawing a picture really kind of makes you remember what you did.
Yeah, I understand which one, like sympathetic versus norepinephrine. I understand that part, but I just get confused of like when like the post starts. And you know what I mean?
Yeah.
Just like when I guess to write it to where it makes sense.
Well, for both of these systems, there's always a pre-neuron coming from like the spinal cord or the brainstem, and then it meets a post-neuron somewhere. So whenever two neurons meet, you need to have a neurotransmitter and a receptor. It's kind of the way I think about it. Just like y'all drew on part 1 when you drew those two neurons connected to each other, And most of you drew that on your lab practical. Although I was very sad for a few people that only drew one neuron and labeled it perfectly. I had to take points off because they didn't draw two neurons connected to each other. I was actually pleasantly surprised. Most everybody got all their points for that question. The lab practical. All right, what else?
Can you go over the reflex arc?
One we just did.
Yeah, can you just explain it a little bit better?
Yeah, I'll just use it off the chart. So basically the heart rate reflex arc is you have a sensory receptor that senses the blood pressure. And then it has an axon that goes to the medulla oblongata. And then the medulla sends, well, I guess the medulla decides what to do about it. And then it sends information, either activates sympathetic or parasympathetic nervous system to fix the either low blood pressure or high blood pressure.
Do you know the steps in order, or just kind of overall what happens?
Well, I mean, you should know it in order.
Well, yeah, but I don't know. Like, if you tell me to do it one way, I'll learn it. I don't know.
I mean, it happens in order. You know what I mean? So,
If I think of having to like memorize a list of things, it confuses me more than just knowing the
yeah, well, I think the way to think about it is, you know, how does the medulla know which system to activate, right? It needs to sense what the blood pressure is. So the blood pressure sensing has to come first. Which I think makes it a little easier to remember if you think of it that way.
So it's basically just reading blood pressure, the axon, the medulla, or that sends getting sent to the medulla, and the medulla taking the fan high or low, and regulating it that way.
Right. So the medulla either, if the blood pressure is low, which system do you activate? Blood pressure is low, you activate sympathetic. And if blood pressure is high, it'd be the parasympathetic, right?
People that have like chronic low blood pressure, is that just like, does that mean that they're just not getting, it's not getting sent for whatever reason, or like there's not enough?
It's probably just some weird genetic thing, if they don't actually have an underlying disease or something. Because, yeah, there's, you know, blood pressure is largely genetic, and some people just have lower blood pressure than usual. I don't know why that is exactly. It could be the medulla deciding that blood pressure is fine. We don't need to change it. But that's on part 2. Sure, so let's pull up part 2 here. So on our nervous system diagram, so the signals from the heart, or from the brain to the heart. So we've got, so basically I'd say brain, brainstem, so brain, brainstem, and then it's going to come through the vagus nerve to the heart, basically. Yeah, so it's still, so from the brain to the brainstem, through the preganglionic neuron, which goes as part of the vagus nerve, and then it meets the postganglionic neuron at the heart, basically.
So what it's like that you just
Yeah, so as long as you can describe that, what that chart or diagram means, oh, it'll be pretty easy. And as a reminder, for all of these, if you don't tell me which about the preganglionic and the postganglionic neuron, you're not going to get all your points.
So do you want us to write that before and say the next step here?
As long as I know that you know where the preganglionic neuron starts and ends,
So we can like put in like,
you could put it in separate sentences. So you could say like the signal goes here to here, and then yeah, the preganglionic neuron is from here to here, and the post is from here to here. That'd be fine.
From the brain to the heart, it's the preganglionic and the postganglionic meet at the nervous nerve, right?
No, they meet way over here at the heart.
Oh, because that's right, because the parasympathetic, the postganglionics are always like in the organ,
Mostly, except for these ones up by your eye and stuff.
but your visceral organ.
Right. Exactly.
Postganglionics are always in.
Right. Right. And you don't necessarily memorize that because you'll have this in front of you to look at. So, and it's even got a key on the top, right? That's the pre and the post. So, so as a suggestion then, what do you think you should have out while you're studying chapter 15? Should probably be looking at your chart when you're studying it because you'll have that with your test. So the more you look at it and think about it, the more helpful it is. I can't forget. I know I'm going to forget to print those and someone will have to remind me. Anything else from 15? F from part 1? I figured someone asked about that one. All right. Let's see, which one makes more sense? Where'd it go? Wish I could have it at the same time. All right. So for this one, you did kind of work on this on that worksheet on part 1 where it said like find a signal that does this, one of these 4. So first let's go over, we can just go over which kind of the 4 ways and then we can look at it on the diagram if I could find one. All right. So the first thing is kind of the easiest. So an axon may synapse. Oh, maybe first we go over this. So this is the 4 ways that a pre-neuron can meet a post-neuron in our sympathetic nervous system. So we've kind of put them into 4 categories. The first category is where a preganglionic meets a postganglionic, basically right at the first ganglion that it meets. So if we look at sympathetic, that might be one that meets at the sympathetic trunk. So like the ones that are going to the lungs, for example. As you could see, the ones that are coming from the lungs, there's the pre-neuron. It meets the post right when it gets to the trunk. First ganglion it reaches. It doesn't do anything else. It just goes there, and there's the synapse that goes to the lungs. So that'd be like the first possible way. We also have an axon may continue without synapsing through the trunk to end at a prevertebral ganglion. So is there anything on here where it goes through the trunk without making a synapse? So that'd be like any of the dark lines that just went straight through the trunk. because that means if you didn't become dotted, you didn't have a synapse. So all these ones going to the intestines, it came from T5, went straight through the trunk, and it met a ganglion there. That'd be the second way.
Okay, so like the ones from the trunk to the heart wouldn't be because they switched the
Well, the heart's a confusing one because it's got two different kinds. So, but let me talk about the heart next. So, well, 3 is easy. There's actually only just one. So that's the axon, the preganglionic neuron that goes all the way to its target organ without any synapses.
What's the pre-vertebral ganglion?
Yeah, so those are these blue ones, the blue circles.
Does that just mean it's outside of
It's outside of the trunk.
the trunk? That's all that is.
And so there'd be like a cluster of connections there.
And is a ganglion like, I don't know, I think about it this way, but this might be wrong. This might be what I'm confused with. I think of the ganglion like it's the beginning of the nerve. But,
I'd think of it more as where a bunch of neurons meet each other.
okay. So the nerve is technically just the axis.
Right. Yeah, I wouldn't think of it as a nerve.
Okay, so it's not, okay.
That's right. But it's a location where a bunch of nerves meet each other and exchange information. So anyways, number 3 was the preganglionic neuron goes straight to the adrenal gland. So that's the only one where it's a solid line all the way to the target tissue right there. And then the last one was this one. So the axon either goes up or down the trunk before it has a synapse. But the synapse is still in the trunk. So do you see any axons that do that?
From the bottom to your eye.
So the ones that go up to your eyes, that'd be true, because they started like at T1, and then they went all the way up and had a synapse higher up on the trunk. Because we had our solid line going from here to there. And that's one thing this diagram doesn't show very well. But anytime you see this solid line here represents any neurons that started there and went up. Same thing down on the bottom, like the ones that are going to the bladder, they came here, the pre-neuron, and then it came down the trunk and then had a synapse. So that'd be the 4th way. So once again, if you know what that diagram's talking about, do you think you could figure out the 4 ways without memorizing them? Or at least get a few of them, get 3 of them maybe? Haven't decided. We'll see. All right, anything else from part 1?
Yeah, for B, it's actually A, it's like only dual and
Yeah, so the definition of dual innervation means,
I just don't understand it.
yeah, I'll keep it simple. So it's an organ that has connections to sympathetic and parasympathetic.
So if we were all close,
Well,
they're on.
a lot of them,
Yeah.
but I mean, you could certainly figure it out by looking at it. But yeah, just that definition means it's both. So like the heart, it gets input from both systems, so it has dual innervation. Sure. You want to go back to chapter 12? Since we got 25 minutes. Didn't bring it? All right, so chapter 12, part 1, asking about G. What did you get for G? So G is, the 3 factors that maintain a resting membrane potential in a neuron.
Yeah, I put a large amount of negatively charged protein and phosphate ions.
Inside.
And then lots
So 12 part 1, G. 3 factors that maintain a resting membrane potential. We have large amounts of negative proteins and phosphates inside.
and then I put lots of potassium channels and few sodium channels.
Right?
And then just the NA.
Yeah.
And then
So that's good.
okay, I just want to know.
So we've got so we got the first one. The second one was lots of potassium channels and few sodium channels. And I think if I bring up this picture, that might help. And then the third is the sodium-potassium pump, because sodium-potassium pump pumps out more positive than it brings in to the cell.
There's large amounts of negatively charged particles, just like proteins and phosphates.
Mhm.
What's the other one?
The other two, sodium-potassium pump,
No, but there's just, so proteins and phosphates is enough, just as like,
that's one.
as negative charged particles. And
That's
then-
one answer.
And then, so lots of sodium, potassium, and
And then lots of potassium channels and few sodium channels in the membrane.
I just didn't know if I had.
Yeah, well, you got it. Which one? The 6 types of Did we even do that? Maybe we should just take that one off. The 6 types of sensory receptors. What?
I said not in class.
Yeah, we didn't do that in class. I wouldn't worry about it. We just ran out of time. I don't even think I put it in the PowerPoint. I think I forgot that was on the study guide.
Yeah, it was kind of like, well, it's kind of like, we can just kind of remind them, so I didn't think we really had to go over it.
Yeah, but it's okay. I think you all have enough. I'd rather ask you about neurons and action potentials than sensory receptors. So that's the last one from study 12, part 1, to cross out. Nothing else from part 1? I guess part 1's easier than part 2. Part 2I. Contrast a neuron's response to an excitatory. Is that how you say that word? Excitatory versus inhibitory neurotransmitter. So right there in the name, it kind of tells you what they do. So which one do you think is more likely to start an action potential? Okay, and to start an action potential, what kind of ions do you let in? So you could say sodium, could say positive. Because it's both basically true. So if excitatory lets in positive ions, what do you think inhibitory does?
Or like what are examples? Is it like the phosphate or proteins?
It'd be letting in negative ions,
So like what are the negative?
like chlorine or chloride, Cl minus. You could let those in. But yeah, basically excitatory lets in positive ions, which is going to get your membrane potential closer to threshold. And then the inhibitory is going to do the opposite. Let in negative ions, get you farther away from threshold.
So on E, I only got 2. I put sensory motor and
You mean the 3 neuron types that make up a somatic sensory pathway? It's primary, secondary, and tertiary. Or wait, no, wait. Or is it first order, second order, third order? Yeah, that's probably how I taught it. First order, second order, and third order. And so basically, you know, the first order takes it from the, you know, out in the body, peripheral nervous system, to the central nervous system. And then the second order goes from, you know, like the spinal cord to the brain. And then the third order is from one part of the brain to another part of the brain.
What was the first one?
So it's from, you know, like the peripheral nervous system to the spinal cord. Second order is spinal cord to brain. Third order is brain to brain. I think it really should probably just say the 3 neurons or something. It's kind of a little confusing the way it's written.
So, can we just like narrow it down? Because I have a lot written from the book or from those slides.
So how an action potential is propagated down an axon? Who thinks they have it written simply?
When it reaches threshold, when the neuron reaches threshold, it continues to open the gate.
Why don't we go back to H to talk about how we started an action potential for part 1, and then we could, on part 1, and then we could talk about how it goes down the axon.
See, that's just kind of what I wrote.
All right, let's try. All right, so if we're talking about how you generate an action potential, first let's make our pretty little neuron here. We'll just give it one sad little dendrite. So this is the axon. Here's the cell body. All right. Let's see if we can. So green. Did I just put green on my face? That would have been funny. Okay, so green will be our ligand gated channel. And then in blue, try to make some little channels here. Or blue, red, voltage gated. And I would say if you're feeling like you're really struggling with this part, most likely it's because you're confused about how the ion channels work, not about the process. So you want to go back to that ion channel part, you know, recompare and contrasted them, maybe look through the PowerPoint about, because all it is what opens them is different. So what opens the ligand gated channels? Ligands, which are usually chemicals, some kind of chemical. Positive ions do not open ligand gated channels. Neurotransmitters.
Because I have in like parentheses,
Well, so what happens when the channel opens? It lets in positive ions.
but what is it that opens neurotransmitters?
Neurotransmitters, you could just say ligands or chemicals. What opens voltage gated channels? Change in charge, change in membrane potential, change in the amount of positive ions that are close to it. All three of those things mean the same thing. And what do they do? What do they let in when they open? Sodium, right? So I think that's where people actually get stuck, is how these channels work, not so much about the whole process. So you want to make sure you've got those channels, how they work, before you try to understand how the neuron works. All right, so anyways, in our dendrite, we've got some ligand-gated channels, and we've got some voltage-gated ones, the cell body too. The axon has them, and if you notice, I'm drawing them all closed, or trying to. Whoops. All right, so all our channels are closed to start with. And then the inside of the cell, is it negative or positive?
Negative.
All right, so negative, 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 all over. So let's say some neurotransmitter thing comes by, binds to that channel. What happens to it? All right, so it's bound. Let's open it. Open. And perhaps it's a sodium channel like most of them. So what's going to come in? So all those positive sodium ions that are outside, they can come in. What does that do to the voltage-gated channel? It's going to change membrane potential and cause that one to open. So it's open, which is going to let more positive ions in. Moving in. What's it going to do to the next one? That one's going to open, let more positive ions in, and the next one, right? So that's what happens to start an action potential, and then the positive ions opening the next voltage-gated channels is traveling down the axon. They all just react to the positive ions coming in. So we open this one. Those positive ions make this one open, which lets in more, which opens this one, and so on and so forth.
So as far as explaining the propagation, it's just you can just simply write once an area of the cell reaches, once it crosses its membrane threshold, then it just moves, the cell becomes more and more populated, so it causes channels to open
Right.
all down.
Right. And in terms of like this picture, this part here is generating an action potential, and all the rest of it is propagating it. So like that's the generating part. And then everything else, you know, this part is the propagation. What happens at a chemical synapse when the action potential gets to the end? What is the axon terminal release? So neurotransmitter. So that'd be like, you know, at a chemical synapse, you get that action potential, and then it signals the terminal to release the neurotransmitter, like acetylcholine or norepinephrine, to activate the next neuron. I mean, synapses are in every chapter, I think. No, it was in 12. Chemical and electrical. I don't know, do you have any more questions on 12? Part 2. Going once, we got 10 minutes. Going twice. I'm gonna go to 13.
Can you go with the plexus? Not really what each one does, but just overall.
Yeah, so like just basically what a plexus is just a group of neurons that are kind of bundled together that go to one area of the body.
I have one. On G, the spinal cord has, why does the cervical spinal cord has more gray matter than the thoracic?
Oh, and we needed to fix that one, right? Yeah, so just to make sure we have it, should say explain why the lumbar spinal cord has more. So that was G. So explain why the lumbar has more than the thoracic. So it has more synapses because it has to connect to all those nerves and neurons going to the legs and coming from the legs. You can imagine that's a lot of neurons. Whereas the thoracic, there's not as much needs to connect. And also, why do you think there's more white matter as you get higher up in the spinal cord? Okay, there are more axons. Why are there more axons there? Mm-hmm. Yeah.
Not many places it could have branched off to yet.
Exactly. So like if I look in my spinal cord right here, are there signals going to my hands there? Coming through there? Are there signals going to my feet going through there? Right? So anywhere signals are going are passing through axons in your neck. Right? Because that's above everything. But as you get lower down, if you look down here, you've only got axons, going below there or coming. So I think next semester I'm going to change that thing to ask about white matter instead. I think I like that better. Which parts of the body the plexuses supply nerves to? I think in the interest of time, I would just direct you to the PowerPoint slides for that one. Just because we only got 5 minutes. But there's a slide for each one, and it tells you where each of those plexus go to.
The spinal thalamic, or however, yeah, and the posterior palm, that's just affects like the kinds of touch that you feel basically, right?
Yeah, that was really the main differences. So that was for eye, contrast the spinothalamic and posterior column. The only real differences we came up with were the type of senses that travel through it. So remember, like, one was fast pain, one was slight pressure. Remember all those. Mm-hmm. Oh, for both of those, which direction are signals traveling? Towards the brain or away from it? So it's towards the brain because they are sensory tracts. So they're taking senses that way. Anything else from 13? Maybe you left early, ran away. Or maybe we didn't get to it.
I think that one we only got and then I got sick and didn't come back.
All right. Anything from 14, last chapter?
Does the blood-brain barrier have to do with like the duramater and all that?
No.
Yes.
I thought you were telling me I was wrong.
No.
Throw something at you.
You would like let that slip and get you.
So the blood-brain barrier, and this is where people always get that confused. It's not the dura mater. It's not like the protection of the skull. It's basically all over the brain where the capillaries are. So remember you had like the capillary membrane, the basement membrane, and then the special neuron or special neuroglial cells that wrapped around them. So you might remember which cell that was? So it was the podocyte. Remember that one? Let me look up the picture real quick. Let me see what I actually put on there because I don't remember. Because there's a lot of different terms for it, so I want to make sure I tell you the one I actually put on the PowerPoint.
Why can I not find it now? Blood-brain barrier.
All right, so it's, astrocytes is what we were using. So in the picture you had the tight junctions between the endothelial cells, which is the capillaries, basement membrane, and then the astrocyte feet. And so we've got 3 layers to keep most everything in the blood, not touching your neurons. So you would find this anywhere you have capillaries in the brain.
And then the blood supply of the brain?
Carotid arteries,
Okay.
and then the femoral vein, jugular vein.
Yeah.
Thinking about the leg there.
So that's just, so when you say describe it, you just basically want the blood vessels. The
Yeah.
names of the blood vessels. Okay.
All right. So study well. Email me if you have any questions about any of this.